Good morning, everybody. In this slide, we will talk about phylum Porifera. This phylum, an evolutionary blind offshoot. An evolutionary blind offshoot. The phylum Porifera was coined, and their true animal nature was established by Robert Grant. Members of these phylums are mostly marine. These are they are either radial symmetrical or asymmetrical animals. These are marine. They are radial symmetrical or asymmetrical animals. These are primitive, multicellular and sessile animals. Primitive, multicellular and sessile animals. Their body wall is composed of two layers. Outer is pinacoderm and inner is choanoderm. Outer is pinacoderm and inner is choanoderm. These are cellular level of organization. Cellular level of organization. Next, these coelom. The between the body wall, between the pinacoderm, choanoderm, there is separated by gelatinous matrix called gelatinous matrix called mesohyl. Next, sponges have canal system that constantly conducts water. Water enters through the canal by a minute pore called ostia. The water enters into the body canal through the minute pore is called ostia, and the body wall into the central cavity, the spongocoel. From where it goes out through the osculum. The water goes out through a osculum. These are excellent apparatus. The transport of water helpfuls gathering the food and respiratory exchange and removal of waste. Choanocytes or collar cells. Collar cells are lined the spongocoel and the canal. Choanocytes and collar cells line the spongocoels and canals. Nutrition is holozoic and digestion is intracellular. Sexes are separate. The nerve cells and sensory cells are absent, so they do not show much coordination between the functioning of various parts of the body. So next one is the body is supported by a skeleton made up of calcareous or siliceous spicules or spongin fibers. Sexes are not separate. Hermaphrodites are monoecious. Eggs and sperms are produced by the same individual. So sponges reproduce asexually by fragmentation, budding, and sexually by formation of gametes. Next one is fertilization is internal, cleavage is holoblastic, and development is indirect, involving different type of larval stages such as parenchyma, trichymella, and amphiblastula, which are morphologically different from the adult. Power of regeneration is well developed. They are considered an evolutionary blind offshoot. Examples are Sycon eupluctella, and Spongilla and Euspongia. These are the examples of poriferan animals. Those are Sycon eupluctella and Spongilla Euspongia. So classification of phylum Porifera classes classified into three classes. Those are the Calcarea, Hexactinellida, and Demospongia. Calcarea, Exoticnellida, and Demospongia. Calcarea. Examples of calcarean animals are Sycon, Leucoselenia, and Grantia. Sycon, Leucoselenia, and Grantia. Examples of class Exactinellida are Euplectella. Euplectella, also called Venus flower basket, and Hyloneema. Hyloneema is rope sponges. Next one is Demospongia. Examples of Demospongia is Spongilla, freshwater sponge, and Euspongia, bath sponge, and Chelina, dead man's fingers. Dead man's finger. This is about of some general character of phylum Porifera. Fori means bearing four. Their body wall, total body wall bears some small pores. Some small pores. This name was coined by Robert Grant, and true animal nature also established by Robert Grant. They have developed water canal system through their between the pinacoderm and choanoderm. There is a gelatinous matrix. Water enters through the ostia and exits through the osculum. Okay, these are this canal, and coelom was lined by a choanocytes or collar cells. Sexes are not separate. Some sexes are not separate. Next one is produced different individuals. Egg cells and sperm cells are produced by the same individuals. Reproduce asexually by fragmentation, sexually by formation of gametes. These are the different type of larval stages: parenchyma, trichymella, and amphiblastula. Okay, thank you.